Hello, good evening to everyone. Today we will learn the cloning and the role of gene actions and cytogenetics in farm animals. That means the simple terms what we can say is we will are going to learn what is a cloning and how it is applicable to the farm animals and animals. So it's a comprehensive study of cloning, gene actions, and the cytogenetics in the context of the farm animals, maybe a cattle, sheep, goat, and pig, and many more. So cloning involves the creating the genetically identical copies of the animals. Then the gene actions influence the traits, while cytogenetic studies chromosomal behavior and the structure. So these aspects are critical in improving the livestock sector. So because of this thing, the aspects are the critical in improving the livestock breeds for productivity and also the welfare of these particular animals. So, cloning in farm animals is a biotechnological tool that is used to replicate the organism's genetic material in toto. So, it is primarily aimed at reproducing animals with desirable traits like very high milk yield, fast growth rates, and disease resistance. So, this is the main aim of the cloning is concerned. Whereas types of cloning are the embryo cloning is number one, which involves dividing a developing embryo and implanting it into the surrogate mothers. So this is the embryo cloning. Whereas somatic cell nuclear transfer or SCNT is another type wherein which a nucleus from a somatic cell is transferred into a enucleated egg cell, creating a genetically identical animal. Then somatic cell nuclear transfer is gained attention when a first sheep dolly is produced by this particular technique. So the technique allows the cloning up of adult animals with very specific traits, making it a powerful tool for livestock breeding. Hence, this drawn the attention of entire world. So, what are all the applications of the cloning? So, cloning is used for reproducing elite animals, conserving endangered breeds, the biopharmaceutical production, and research into the genetically originated disease. So, the reproduction of the elite animal is one of the challenging thing because the elite animals means having the superior traits or the qualities in terms of the production of the milk and also the egg or sometimes the wool, etc. So, the conservation of the local breeds is also another application of the cloning, which will surely help to conserve endangered and threatened breeds by producing genetically identical animals. For example, the Punganur breed is almost extinct, and if we get a single Punganur breed, then we can genetically modify it in such a way that a cloned Punganur cattle can be generated. The application of the cloning in the biopharmaceutical production is the cloned animals are used to produce therapeutic proteins in their milk, enabling large scale production of difficult to synthesize the biopharmaceutical productions. Then R&D. What is the role of this is for studying some of the genetic disorders which are inherited from the parents or sometimes acquired by. So this can be used by the genetically modified animals and genetically identical population allow for controlled studies also. So gene actions in farm animals is another big topic where we can describe how genes influence traits like the growth rate of a broiler, milk production in a dairy cattle and the disease resistance in almost all the animals. So, the understanding the gene actions is essential for genetic improvement of the programs. So, what are all the types of the gene actions? One is the additive gene action, then the effects of individual animals. Genes accumulate, affecting the traits proportionally. So here the activity get added. Whereas in case of dominance gene action, the dominant alleles mask the effect of the recessive alleles. So both of these are existing. Then epistasis, it's a process of interaction between genes. Then that influence the trait of unpredictability. So additive gene action, so the traits like milk yield are influenced by selective genes with additive effects. So selection based on these genes leads to gradual improvement. Then dominance action here the one of the gene allele is dominant and it only expresses and suppressing the other dominant things. So in animal practice, we need the dominance and also the recessive properties. Whenever we require a docile animal, then we can incorporate a docile gene into the next offspring. Then epistasis in farm animal is involves the interaction between genes that affect complex traits like growth rate, disease resistance, and in the unpredictable ways which are genetically inherited one. So cytogenetics is also another important tool for farm animal. It's critical for understanding genetic makeup and identifying the chromosomal abnormalities. So that's why the genetics is mainly focusing on the chromosomes, but now the era has been changed. Usually, the chromosomes should be equal in number and size, especially in case of the male or either male or any other the parent. So the farm animals have specific chromosomes number like the 16 case of cattle it is 60 and 57, then pigs 38. Then these deviation can affect breeding potential and productivity, which is originally evaluated in the natural way. Then karyotyping and chromosomal analysis is one of the process in which the visualization of chromosomes to detect what is the abnormalities or translocations and also which affect fertility and the development. Then the role of genetic engineering. We are geneticians in the uh, cloning is enhancing the breed's herd capacity to produce very high yielders only and the culling the other bulls etc. Then the integration of cloning into the cytogenetics is already being done, and these are all targeting the genes which are responsible for the disease, maybe in the source. Then marked resistance selection, it is selected to a specific traits of accurate selection. The cloning replication are superior in case of animals and rapid trait also. So, what is a genomic selection? Its gene editing means the CRISPR. So modifies genes from abnormally against abnormality and also the economically important diseases. So the cytogenetics analysis monitors the genetic makeup of these animals. So cloning and cytogenics are already internally related and they are very stable in case of once they are incorporated. But there are a lot of ethical consideration. Then the nowadays for simple experiments also we need to take so many steps 1 by 10. I can list it here, but it's not the forum. Then cloning the path is a type of the cells or certain part of the cell or DNA from a parent cell to propagate a certain desirable characters. That's why cloning is popular. So three types of the cloning. One reproductive cloning or main focus. Then second one is the gene cloning, and the third one is the therapeutic cloning. So varieties of these interesting factors are there. Let us go by one. In 1885, first, first ever demonstration of artificial embryo twinning has been shown by some of the scientists. Then they might have done the research and ultimately published the papers. So 1902, the artificial embryo will not bulk. It's not possible. So 1918, then a cell nucleus controls the embryonic development was now and Cox Nexus successful nuclear transport has they happened in 1952. Then 1958 nuclear transfer from a differential cell is also another entity here. Then in the 1984, first mammal created by nuclear transfer. 1987, nuclear transfer from embryonic cell is also is generated. So these are all the chronology of the events, how it went well. So we know that a somatic cell or the egg cell or the sperm cell, how they are looking ahead in the electronic microscopy, the somatic cell is shown there. Then somatic cell this clear transfer is SCNT. Then one need to occupy this thing, then guaranteedly. So we'll see. So somatic nuclear transfer, how it is transferred is it's a big journey. Suppose a sheep or any animal farrows, or it is going to have the child. And if you study this particular dolly, you get much more idea how the DNA and RNA virus are having some difference, and they are unnecessarily dragging the things. AI is also separate ಮಾಡಿದ್ದೀವಿ that but it is incomplete. The mechanical separation of the cells is already under progress. Then artificial twinning, how to do? So a single fertilized ovum will be injected with the semen. Then there will be an agreement that he or she has to give a lump sum amount. So all this led to find a very cheap technique to get acquainted with the artificial twinning. So this is the twinning process. Separate nucleus will be followed. If it is visible, then surely you can meet it. Then the artificial twinning also says that 90 micrometer is the highest size. So artificial twinning already you have very well experienced. Twinning is a process to a cow when which at the same time it is going to give birth to the new offspring. So dolly is one of the animal which is cloned by the scientists. Then Pimulina, the mouse, has also been cloned by the Japanese scientists. Sample cloned animals, the carp is also one animal. Then the mira, the goat or the cows also do suffer from the particular disease. Then Millie, Alexis, Krista, Doctum, and the Karen are the names of the sample cloned animal. So Umbretta, the Mouflon, is a breed that is evaluated out of the pregnancy PMSG induced estrus. So usually the cloning is always done with the monkey, rhesus monkey or macaca mulata, what you say. Then also it can be conducted in the no or gar animals to get the better production and reproductive performance. So Ralph is the rat of the many YouTubers. Libby and Lily ferrets. So North Korean clones first human baby was generated by this thing, and can we clone to humans? The questions are multiplication of animals by cloning. Then the technical medical safety is will not be there as it is not in the controlled condition. Then what is the benefits of the farm animal cloning? So it also makes us to retain the original genetic trait of that particular animal. Then it reproduces the healthiest animals, thus minimizing the use of the ABs. Consumer benefit from cloning because meat and milk will be most helpful, consistent, and safe. Then the animals which are already endangered or their population may be vanished from the earth, such animals can be cloned. Then transgenic animals is one of the recent concept wherein developing these cloning techniques to facilitate the genetic engineering of the animals. Bioethical issues are also there. Many issues are there. We need to address all this, taking that the benefits are more when compared to the risks are concerned. So human cloning, of course, the Japanese Council for Science and Technology, it adopted it, may lead to breeding of human being and violation of the human rights. So these are all some arguments, and the legal entities are also instructed. The November 1991 onwards, we need to take something, and hope you might have put some questions. Molecular cloning, cellular cloning, and embryo cloning. Then clone is the replica of the original animal, and in case of animals, it is somewhat less used as the cost of treatment is very high. So we at this moment we have to wait and see. It is a non-sexual production of the offspring animals. No fertilization, nothing. Then the Hans Derich separated the blastocysts of embryos in sea urchin. Then later it went on several times. Then, depending upon the unavailability or efficient handling of the system, which can be combined together with that of a reputed persons who do have the interest. So, lack of regional that mammalian oocytes and embryos require strictly controlled temperature for development. So many it begins at 1980s or earlier, and nowadays everyone is noting the cloned animals. And in very old days, the embryologists declared that the cloning up of the mammals is impossible. Near nuclear transfer was biologically it's impossible, but later it was proved that it is possible. So there are early success were also dealt like in 1986, cloning technology was successfully established in India. Then some techniques were successfully used by the goat or sheep farmers. So embryonic cells, etc., can be used in future in case of the human beings because lot of research is going in the human field. Already we know that a CNT, etc. Then remarkable things. Here the steps in cloning is we need to collect the ovaries, then in vitro fertilization to be more, then inoculation of the oocysts, then the swing, then fusion of the somatic cells is also another word, the embryo transfer into the recipient. So cloning involves the procedure of searching a donor animal. Then we are going to take his soma or body cells. Then the donor vehicle will get such type of the erythrocyte. So recipient cytoplasty. Then other techniques have not worked here. Application of cloning. What are all these things? Number one, it is the trend, common trend. And if you ask any student, they will say that they will join the Public Service Commission department and they can arrange wonders. So it's also used as the tool, especially in case of biomedical engineering, and you can see already the genetically proper animals. They are not going further, whereas the other things still it is not started in human beings. We do not know. We started. So just go through these operations already several times. You have been learning this thing. Stem cells research is also another technology that is evaluated out in Bangalore. So there are problems also development of anomalies, short-term lifespan, large offspring syndrome, then aging up of the animals, then she may not find us. So these are also some steps systematically the department of dairy, then also the dairy industry. But there is one myth that the early death of the doggy died due to, then doggy died because of the retroviral disease. But almost all the cloned animals have a normal lifespan when compared to the usually the average human lifespan. Large aspirin syndrome. Then there are a lot of causes are there which need to be detected if he or she is reluctant to send. So this is ultimately what we can conclude is cloning gene actions and cytogenetics are the one integral part of the animal genetics and breeding, and these are very crucial for livestock breaking. And these techniques used responsibility then enhance productivity while considering the ethical complications. So that's the end of the cloning.